Imagine the human cell as a city of trillions of tiny machines called proteins. Most of the time, these machines work in perfect harmony, but sometimes a machine breaks. It starts producing too much of a signal, or it blocks a vital pathway. This is where disease begins. For a century, our strategy to fix this was simple: occupancy. If a protein was causing trouble, we built a molecule to act as a plug. We called these inhibitors. Like a key in a lock, the drug would sit in the protein's active site and freeze it in place. This approach gave us aspirin, statins, and the first generation of cancer drugs. It felt like we had the city under control, but as we mapped the human genome, we discovered a terrifying truth. Most of the proteins driving the most deadly diseases like certain types of cancer or Alzheimer's don't have locks. They're smooth, flat, or completely unstructured. We call these undruggable. For decades, these proteins were the ghost cities of medicine. We knew they were there, we knew they were killing us, but we had no way to touch them. To understand how to defeat the undruggable, we have to stop thinking like chemists and start thinking like the cell. Because the cell has a secret, it doesn't actually need a pocket to destroy a protein. It uses a system called the Ubiquitin-Proteasome system, or UPS. Think of the UPS as the cell's waste management department. It consists of two main players, the tagger and the shredder. The tagger is an enzyme called an E3 ligase. Its only job is to patrol the cell and find proteins that are old, misfolded, or dangerous. When it finds a target, it glues a tiny protein called ubiquitin onto it. This isn't just a label. It's a death tag. Once a protein is tagged with a chain of ubiquitin, it is recognized by the shredder, the proteasome. The proteasome is a massive, barrel-shaped machine. It grabs the tagged protein, unfolds it, and rips it apart into tiny peptides. It is the most efficient recycling system in the known universe. Now, here is the breakthrough. To scientists, Craig Crews and Ray Deshaies asked a radical question: what if we could force the tagger to label a protein that it would normally ignore? They realized that if they could build a bridge between the E3 ligase and the undruggable protein, the cell would do the hard work for them. They wouldn't need to block the protein, they would just need to flag it for execution. This bridge is what we call a PROTAC, a proteolysis targeting chimera. A PROTAC is a molecule made of two different heads connected by a linker. One head is designed to grab the disease-causing protein. The other head is designed to grab the E3 ligase. When the PROTAC finds both, it pulls them together into a ternary complex. It's a molecular matchmaking service. This is where the magic happens. The E3 ligase, now brought into direct contact with the target protein, transfers the ubiquitin tag. The PROTAC then releases both and floats away, completely unchanged. This is the fundamental difference between old drugs and PROTACs. A traditional drug is like a soldier who has to stand guard over a protein forever to keep it quiet. This is called occupancy-driven pharmacology. But a PROTAC is like a hitman. It enters, marks the target, and leaves. One single PROTAC molecule can mark, destroy, and then move on to mark another target, and another, and another. This is event-driven pharmacology. It is orders of magnitude more powerful. But the road from a lab bench to a pharmacy is rarely a straight line. In 2001, when this idea was first published, the world wasn't ready. The first PROTACs were made of peptides, large, fragile chains of amino acids. To a chemist, peptides are a nightmare. They are too big to cross the cell membrane, and the body's enzymes chew them up in minutes. The consensus in the industry was clear: this is a beautiful biological trick, but it will never be a drug. For years, funding was scarce. The idea was relegated to the fringes of academic curiosity. But Craig Crews and Raymond Deshaies refused to let the idea die. While Deshaies provided the biological roadmap of the cell's shredder, Crews fought the battle of the molecules. Today, The impossible is happening. Small molecule targeted protein degraders are in human clinical trials, tackling cancers that were previously untouchable. The companies born from this idea, like Arvinas, are redefining how we treat disease. But this is more than just a new drug class. It's a new way of seeing the body. We are learning that we don't have to fight the cell's machinery, we can partner with it. We are no longer just plugging holes. We are cleaning the city, and we're just getting started.